Hi, good morning to everyone. We are continuing to discuss about the molecular basis of inheritance. Around 1953 to 58, around this time, people already knew that DNA is the material, or DNA is the genetic material that transfers from one organism to other organism. That was known by the work of many scientists. That includes Watson and Crick, that includes Griffith, Hershey and Chase. All these people proved clearly that DNA is the material that is going to be transferred from one organism to other organism. When Watson and Crick suggested the model, they also mentioned that DNA has got the semi-conservative technique of duplication, but this was not proved clearly. At that time, there were three important ideas were discussed to explain about the replication or duplication of the genetic material, because one parent used to pass on the information to other cells, and they wanted to know how. They knew there is a replication, but how it was happening was not known. So three postulates were there. One is called the conservative type. Second is called semi-conservative type. And the third one is called dispersive. So these were the three important ideas that were prevalent at the time. Many scientists were trying to find out which is the correct one, but I think in any of the science experiments, luck and selection of material plays a very important role in coming out with the result. It was at that time MESELSON and STAHL. In 1958, they published a paper. These people were working in Caltech, that is the California Institute of Technology. They were working on that material. So they were very intelligent to select the materials for their experiments. What they did was they took a organism called E. coli, a bacteria that has got a very short life cycle, that is probably in about 20 minutes of time that will be able to have one cell cycle. Cell division will form in 20 minutes in the lab condition. That's the first thing they selected. And in the second one, what they did was, they at that time, not many biochemical methods were available for analysis and study. Because today when we want to separate the segments in the photosynthetic experiment, we use the paper chromatography or electrophoresis and all these things. At that time they were not known, so they had only one option, and that option was to using the centrifuge combined with another system of using what's called the density gradient analysis. That means this particular thing, for that they used a material called cesium chloride. Cesium chloride is a material. This material, when centrifuged and allow it to settle down, supposing this is the after centrifuge they do it. When once they centrifuge and do it, this will form. This will take up the levels in the test tube. That will take up three levels. One level will be heavy and denser material, we can call it, or we can call it as the heavy sediment, or a heavy fraction will settle down at the bottom. And the next one, the little lighter ones. And if I say it is the bands, I will indicate in the form of the little lighter one will be in the middle. and still very light one will be in the third one. So if you look at the whole thing, based upon the density, that is the density, very dense one will settle down at the bottom, something less than that will settle down in the middle, and lighter one will settle in the uppermost thing. So these are the three different types of banding or levels we get it. One is the heavy at the bottom, second is little lighter, and third is light. So this was also a technique they used for their activity. And the third one, what they selected, was also required because they wanted to find out which is heavy, which is light, because you know very well DNA and all the proteins in the system will always contain a nitrogenous base or chemicals having nitrogen. E. coli also has the nitrogen in its DNA, and that will be a normal nitrogen with N14. What do you mean by N14? N14 refers to, it will, in the nucleus, it will contain 7 protons and 7 electrons, and that becomes 14. And there will be one more form of the nitrogen, and that nitrogen will have 15 as the mass number or its molecular weight or atomic weight, whatever you call it, and that will contain 7 electrons, 7 protons, 1 neutron, and that becomes N15. These experiments are highly sensitive, and even the slightest change will be indicated. So therefore they took the two types of nitrogen. One is the N14 and second is the N15. So these were the materials they selected for their experiment. Now let us go for the experiment what they did. So what they did was, I think what I do, I will rub it off. I will just rub it off and then go to the next part of it after understanding. So we now know that they selected E. coli, which has got a very short life cycle. They selected the technique of density gradient analysis for analyzing the results, and they thought they took two types of nitrogen. One is the normal nitrogen containing 14, and another is 15 containing what they call it heavy. So they called it heavy. So these are the three materials they took, and what they did we shall now try to find out in a very simple way. Though the experiments took of years and years, we will study that in only 3 to 4 minutes. What they did was they took the regular E. coli, and this E. coli was made to grow in N15. I am using red color for the analysis, therefore I will mark it as N15, that is the heavy one. So they grew it for a long time, and that is what I referred here as the G1. In the G1, that is generation 1, was grown on N15. So this is not a natural one, the experimental condition. They grew, and then they analyzed it with the help of the density gradient analysis. They studied. They found out almost all the fraction settled down at the bottom, indicating they were heavy. So that's the first thing they got it. And then what they did was they took this G1 and then they removed this 4, then they made it into 14. That is the light one. So the second generation was grown in the lighter nitrogen. They analyzed the results after growing several generations were allowed to grow. They took the E. coli, they centrifuged it. When they centrifuge, the cells will break apart and they release the DNA material, and that DNA material will be used in the centrifuge with the cesium chloride and analyzed the density gradient. At that time, they found they were in the middle. Neither they were heavy nor they were light, but they were in the center. So this was the type of experiment they were doing it. At that time, they found out if it is the conservative type. What is the conservative type? They would explain. In the conservative type, they say that the DNA material that is present in the parent somehow will make a copy by itself into two. That means they keep on this helical structure conserved. The parental DNA conserved as it is, and then they make the copies. How, it's not explained very clearly, but that is the way it is conserved. The parental original DNA is conserved intact without any change in that. That is one thing. If that is the structure, if that is the one, then I think the result shown here is not correct, because the conservative conservative technique indicates that there will be the original one will be maintained. So that may not be possible in that one. And the second generation, when they took the second generation, they came to a conclusion that the 100% was seen at the middle part of it. That means it was not heavy. So they found that the first one, the conservative, if it is a conservative one, in the second generation, what they did, they should be available. They should be shown at the bottom only. So therefore, it did not happen. Therefore, they said semi-conservative is not possible because there is a change in the gradient. So in the next one was the semi-conservative. So in this one, what is that we understand in that technique was that the original form of the DNA, that is the parental DNA. I'm sorry, I think I will use the one. The original thing was the G1, that is the one that contains the heavy material. And in the second generation, when they treated with the normal nitrogen, then they found out somehow or the other there will be 50, that is it was in the medium. So in the semi-conservative techniques very clearly tells that the parental DNA breaks and then the new strands will be added here. That means when the new strands will come up now, that will come up like this. One will be the parent, the second will be from the N14, the newly added one, and that is called the hybrid. So that gives the answer here. That means 50%, 100% of the material or 100% of the progeny will be shown in the middle one. That means it is neither heavy nor it is fully light. So that is one important information they got it. So then they thought it may be either semi-conservative or it may be dispersive. What happens in the dispersive also, I will just check up. In the dispersive type, the parental DNA breaks here and there, and then the new one will develop according to this one, and ultimately they will form. That means 50% of the parental DNA and the 50% of the new DNA will form. That may be also 50%. So they may be either semi-conservative or dispersive, they thought. That means there should be something in between that. When they took the third generation for analysis, they found out that 50% of the steady material, that is the gradient indicated was completely in the middle and 50% became on the top layer, that is the later one. So now this indicates if it is a mixture of the dispersive type, you should have only one band all the time, but there were two bands. Therefore, even dispersive type was ruled out. So the possibilities are there. If you look at this one and analysis taken, the new, that is the old, that is heavy DNA replicated, and this also replicated. That means we got in the G3 50%, that is 1, 2, 50% with the hybrid type, and 50% will be pure type. So when you take up that one, what do you mean the pure type means? Pure type indicates here with the 14 later one, and 50% will be hybrid type, that is the in the G3. So that is the result they got it. And next they also took the G4 generation and they analyzed that one. When they analyzed that one, again the same thing happened. In the duplication happened, this one duplicates to this one and this is duplicated, and when this second strand duplicates, it becomes on the light medium. Like that they got the fourth generation also was analyzed. In the fourth generation, what they found out was one band was only 25%, that is the middle band, and then the rest of them were all 75. That means 75% was on the lighter aspect of it, but there was no heavy one. That means there was a separation, and the one part of the heavy part was always conserved and passed on to the daughter cells, and the new ones are being added to every new cell. So this clearly indicated that there is a method, a technique where the original or the parental, one part of the parental DNA is completely maintained, and the second part will be added up by the new DNA material, and that clearly indicated it is the semi-conservative type. So ultimately they came to a conclusion that the semi-conservative technique suggested by Watson and Crick is the universal type of DNA replication in almost all the eukaryotes, and they proved that the conservative model and the dispersive model are not correct up to date. So until today, we scientists agree that it is only the semi-conservative model that operates in the replication of the DNA molecule. Because one human cell, a zygote, that is a sperm cell from the father and the egg cell from the mother, will contain haploid number. When they combine, they become the diploid, that is the zygote, and that zygote develops into embryo into human beings that may contain 60 billion, 6,000 billion cells, which will have the same information that is present in the parents. So this is possible only because there will be what's called semi-conservative, means they conserve a part of the parental DNA and they keep on adding the new types. This will give a very good thing, and therefore the scientists called the work of MESELSON and STAHL as a beautiful experiment. This is how it is recorded in the papers. A beautiful experiment of the replication has been suggested by MESELSON and STAHL. We shall stop here for the time being, and next we will study what is the meaning of the RNA world. There is an RNA world hypothesis that explains whether DNA came first or RNA came first. We shall take up in the next session.